Hi, my name is Hannah Morris, and this is my Diego Mastroeni report. Here's some information about the background of Diego Mastroeni. He's an assistant research professor at ASU, and this is some of his research interests. He likes to study healthy aging, neurodegenerative diseases like Alzheimer's disease, single cells, neuroinflammation, and epigenetics. And he has over 40 publications in peer-reviewed journals. The primary article for this report is titled Apparent Intracellular Localizations of H3K4ME3 and how it demonstrates an early epigenetic phenomenon in Alzheimer's disease. As it can be seen, Diego Mastroeni is the first author in this report, as well as working with all of these different colleagues. And this article was published in the U.S. National Library of Medicine. Here's kind of background information for this report. H3K4ME3 is an epigenetic, or a non-genetic influence on gene expression, a mark that is associated with actively transcribing genes in the brains of Alzheimer's patients. The question is, can the process of Alzheimer's disease be identified by the amount of H3K4ME3 in the brain? And the hypothesis is, the amount of H3K4ME3 in the brain can be predicted by the severity of Alzheimer's disease in a patient. The prediction, if the amount of H3K4ME3 is found in a patient's brain increases, then the severity of the stage of Alzheimer's disease is more advanced. For the methods portion, samples of different parts of the brain were taken from deceased patients with Alzheimer's disease. The samples range from stage 1, which is the earliest stage of Alzheimer's disease, to stage 4, which is a more progressed state. Each stage of the disease can be defined by, in a deceased patient, by the amount of plaques and neurofibrillary tangles, or NFT, found in the brain. The more NFT, the more progressed the disease. Early Alzheimer's disease patients will be referred to as ND, or non-demented. They will be used as a control group, as well as patients who did not die due to Alzheimer's disease. The control group will be used to compare findings to. Here is a continued section of the methods. Basically, mice were anesthetized and permeated with a 4% buffered paraformaldehyde. Then 40 micrometer samples of the hippocampus were cut. The samples from the animal brains were incubated in various antibiotic solutions, and these are each given in table 2. It's a really complicated process, but this is kind of a simplified version. The samples were treated very carefully with a variety of chemicals and then used for incubation with human brain tissue. After the human brain tissue was immunostained, the samples were examined under a microscope, as they can be seen here. After the photographic imaging, the cellular localizations of H3K4ME3 were identified and quantified using ImageJ software. These images are shown to the right here. This is a non-demented control group, and as you can see, there are no localizations of Alzheimer's disease here. This is BRCA2 control group. You can see that there are 2 localizations because these people had a very early stage of Alzheimer's disease, so that's why there are only 2. Here's the BRCA4 group. They have a very advanced form of Alzheimer's disease, so there are 3 localizations here that can be seen. So here is the results page. After the human brain tissue was immunized and photographed, it was put into a graph. The graph to the right shows the relationship between Alzheimer's disease and the amounts of H3K4ME3 present. As you can see, there's a positive correlation between the stage of the disease and the amount of localizations found. The discussion, it was concluded that there is a positive correlation between the amounts of cells with H3K4ME3 and the stage of Alzheimer's disease. These findings support the hypothesis and can be used in many ways to identify patients with early Alzheimer's states for earlier diagnosis. Also, these findings give another clue as to what causes the onset of Alzheimer's disease and helps kind of reveal more information so that we can find a way to find a cure sooner. These are the references. This is basically just the article that Diego Mastroeni published. Thank you for your time.